Stanford University.
Our next speaker is Dr. Rachel Wilson, and it's my pleasure to introduce Dr. Wilson to you. Currently, she is the Martin Family Professor of Basic Research in Neurobiology and an HHMI investigator at Harvard University. Dr. Wilson and her research has been recognized by several awards, notably an Eppendorf Science Prize in 2007 and a MacArthur Foundation Award in 2008, and more recently, the 2016 Larry Katz Prize for Innovative Research in Neuroscience. This prize is awarded to a neuroscientist who, like the prize's namesake, is open to new ideas, creativity, and contributes to both conceptual and technical innovation. Dr. Wilson's research concerns the information that sensory neurons provide to the brain and how the central nervous system processes and interprets the information. She has pioneered the integration of genetic tools for neuronal identification and modulation and electrophysiology, and coupling these experimental measures with computational modeling and simulation. Our recent focus has been investigating how the properties of synapses contribute to sensory encoding, and we've just heard from Nico a different take on how sensory information might be represented in the brain. Please join me in welcoming Dr. Wilson to our symposium and enjoying her talk, which is entitled Sensory Encoding in a Small Brain.
So I'm going to focus on actually a small subset of the questions that Nico posed for you. In particular, the question, how do neural channels interact to shape perception and behavior? I can't remember what part of Nico's Venn diagram this actually corresponds to in terms of the discipline I'm supposed to be working in, but we could call it System Neuroscience, which I don't think appeared on your list. In any event, I think it's worth pointing out to begin with that this is an old and important question in brain science. The notable work classically was done by Vernon Mountcastle and colleagues. For example, this plot shows you the response of a peripheral cutaneous mechanoreceptor. Firing rate on the y-axis is shown as a function of the stimulus amplitude on the x-axis. In this case, this is the amplitude of the deflection in the skin. You can see that the neuron's response is very discontinuous. But when human perception was measured in response to the stimuli, they found a very smooth relationship between perception and the stimulus. And so this suggested a simple algorithm by which perception might be constructed by simply summing the responses of many different neurons that have different discontinuities in order to recover a smooth percept. But the algorithm that relates the neural code for, in this case, intensity or amplitude, to the perception of intensity is not always a simple algorithm, and that's suggested again by classic experiments here performed in the 1950s, which I'm schematizing here in cartoon form. The experimental design here was to create a tone cluster, a set of 4 pure tones presented simultaneously, and then to ask human observers to rate the intensity of that tone cluster. And the finding is simple. If the tones are widely spaced in frequency space, then the cluster sounds loud to a human observer. But as the frequency separation between the tones decreases, the percept of intensity decreases, so that now, even though the same amount of sound energy is present, our ears, our auditory system is lying to us and telling us that it's a softer sound. And I think that brings up the possibility that there may be antagonism between neural coding channels that is intervening between the stimulus and our percept of the stimulus. So I think some challenges clearly remain in the task of relating neural activity to perception. Ideally, we would like to understand whether there are general principles that account for this relationship across sensory modalities. We'd like to have an algorithmic description of the relationship between the stimulus and the behavior. And we'd like to understand fundamentally how this algorithm is implemented at the level of neurons and circuits, and how the particular form of that implementation might reflect constraints during development and evolution. So that's a big research program. I'm not going to repay all those promises today, but I'm going to tell you about one particular case study in this problem, and that's the case study of olfaction. I think olfaction is a nice setting for asking this question because the olfactory system quite naturally divides itself into channels. Olfaction begins when odorants bind odorant receptors, and importantly, all the olfactory receptor neurons, or ORNs, that express the same odorant receptor converge their axons on the same compartment of neuropil called the glomerulus in the brain. And then it is hypothesized that signals from different glomeruli are combined in higher brain regions, but we don't understand really the general principles that govern that combination. Part of why it would be important for signals from different channels, different glomeruli, to be combined is that even a monomolecular odor stimulus, a pure compound, will bind to multiple receptors and so drive activity in multiple glomeruli. Because of this, and also because the odor response tuning of different receptors turns out to be statistically correlated, it's actually pretty difficult to independently stimulate different glomeruli with true odors, natural odors. But luckily, in the olfactory system, we can get around this with genetic techniques, because one nice experimental virtue of this system is that there's actually a unique gene for every peripheral coding channel, and that's the odorant receptor gene itself, right? And so that's a nice feature of the one receptor per channel rule. So I want to make the point, too, that this basic circuit architecture is found in organisms that are widely separated in complexity and evolutionary time, organisms like the mouse and the fruit fly. The difference here is one of scale, so there are about 1,000 glomeruli in the mouse, more like 50 in the fruit fly. And I'm about to show you a piece of data from the literature pertaining to larval fruit flies. So I'll just make the point that in the larvae, there are actually 21 glomeruli rather than 50 in the adult. So here's the basic question. How do olfactory glomeruli combine to influence behavior? And this is a question that's been asked experimentally in Drosophila a number of times already. And I just want to tell you about these studies before I talk about the work of my lab, unpublished work on this topic, to give you some context for the problem. So this is a study I like very much from John Carlson's lab. They were studying larvae. You can see here that they've placed larvae in a petri dish. One side of the dish is odorized, one side is not odorized, and in 5 minutes, they count the larvae on the odorized side. And they're measuring here a response index, which just scales with the number of larvae who've chosen the odor. And you don't need to read these individual labels. The point here is that the behavioral response ranges from attraction to aversion, and you get different behaviors in response to many different odor stimuli. So these investigators went in and measured the firing of olfactory receptor neurons, in fact, all the ORNs in the larvae, in response to all these odors. And then they did what amounts to a linear regression. They just regressed the neural data against the behavioral data, and they asked, is there a linear combination of ORN signals that accounts for most of the behavior, restricting themselves to a conservative number of glomeruli? And what they found is that this particular equation accounts for 81% of the variance in the relationship between odor and behavior. And there are a few things that I just want to point out about this equation. So each of these coefficients here represents essentially a fixed and independent contribution of each glomerulus to the behavior. In fact, the assumption of independence is sort of tied in or hardwired into this analytical approach. It's interesting that they could account for so much of the behavior by invoking only 5 different glomeruli, because there are 21 glomeruli in total. And that highlights kind of a problem with this analysis, a problem that Carlson and everyone in the field is, I think, well aware of, which is that I mentioned that the odor tuning properties of different ORN types are statistically correlated with each other. And what this means is that using real odors, you have a credit attribution problem. If the odor tuning of glomerulus 1, for example, is correlated with the odor tuning of glomerulus 12, then you cannot use real odors to disambiguate the contributions, the causal roles of those 2 glomeruli in behavior using this kind of approach. So a different kind of approach gets closer to a causal role. That's the approach of silencing individual input channels. That was used, for example, in this study from Jing Wang's lab. Here they're looking at adult walking behavior in a 2-dimensional chamber, and they infuse vinegar odor into one end of the chamber. The flies are attracted to vinegar. But if you silence one type of ORN, that's one of the 50 channels in the adult, and there are multiple glomeruli activated by vinegar, what they found is that they abolished this behavior, suggesting that this glomerulus is necessary. They also did the sufficiency experiment. Here, they started with flies where most ORNs are silenced. This is, in essence, an anosmic fly. Not surprisingly, they don't respond to the vinegar. And then they rescued activity in one of the 50 channels, DM1, again, the channel that they had inferred was necessary, and they found that this was sufficient, which really suggests that there's only one active channel which is playing a role in this behavior. But somewhat surprisingly, they tried rescuing other channels, and they found that rescuing this channel, VA2, was also sufficient to rescue the behavior, which is a bit confusing. If DM1 is necessary, how can it be that VA2 is sufficient? So I think the problem with this approach is that it's just going to be low throughput by virtue of the genetic techniques that you use. And so it's difficult to disambiguate the roles of different glomeruli. And in particular, it's difficult to critically test the hypothesis that glomeruli are making independent and fixed contributions to behavior. So this is really the question that we're re-examining in the study I'm going to tell you about. And our approach is very simple. We're just stimulating olfactory receptor neurons optogenetically. We're measuring a very simple olfactory behavior, and we're going to compare the behavioral responses to pairs of glomeruli with the responses to single glomeruli. So this slide is just to show you that we can, in fact, recruit ORNs optogenetically. Here we're expressing channel rhodopsin under the control of an odorant receptor gene promoter. I'm showing you ORN firing rate on the y-axis is a function of time on the x-axis. And you can see that pulses of blue light of different durations elicit robust and sustained increases in firing rate. So full disclosure, these blue light pulses are interspersed with red light pulses. The red light has no effect on firing rate as you'd expect. And the reason for the red light will become evident in a couple of slides. So here I'm showing you group data measuring firing rates of 6 different ORN types corresponding to 6 different glomeruli in response to blue light. In each case, channel rhodopsin is expressed under the control of one odorant receptor promoter. What you'll notice is that firing rate grows more or less linearly with the log of blue light intensity, and that it's kind of convenient in this case that these firing rates are overall pretty similar. They're different by only about 2-fold, I would say. All right, so the next task is to then measure behavioral responses to channel rhodopsin activation in these flies. So we allow the flies to walk freely in a transparent plastic chamber. We illuminate them with infrared light from above, and we track their movements with a camera from below. And then we can illuminate them with a blue laser at a time and place of our choosing. And we steer the laser beam onto the fly using a pair of galvanometer mirrors, similar to the mirrors in any 2-photon microscope, like the one that David Tank was telling you about this morning. Not surprisingly, wild-type flies show big responses to the light. They are visual creatures after all, so we make flies genetically blind. We found that blind flies still responded to the light. They avoid it. That we presume is due to thermal cues. So in order to eliminate the thermal response, what we did was to add a second laser, a red laser. And this is just designed to eliminate thermal cues from the apparatus. So at every time and place where we down-modulate the power on the blue laser, we up-modulate the power on the red laser, so that the total light intensity at all times and places within the chamber during all stimulus epochs is the same. And that eliminates the preference in flies that don't express channel rhodopsin. So we give the flies a behavioral choice. Do you want to go into the odor or not? And in this slide, in the next few slides, we're going to be driving channel rhodopsin expression in most ORNs under the control of a gene called ORCO, which is expressed in the majority of the ORN population, so most glomeruli. So this is what the chamber looks like from above. It's a long, narrow hallway. And in all the work I'm going to tell you about today, we make half of the hallway blue, so it contains a uniform intensity of fictive odor, and the other half of the hallway is red, so it's non-odorized. And we've adopted this hallway from some work by Gero Miesenbach's lab. And the Miesenbach lab initially devised this hallway using real odors, and just following their protocol, we pipe air in at the ends and we pipe air out from the middle of the hallway, although we're not actually driving real odors through the chamber. We just carry that forward into our apparatus. So this is what a randomly selected walking trajectory looks like for a fly encountering blue light in this chamber. On the y-axis, you're seeing the fly's position. In the long axis, the chamber versus time on the x-axis. You'll notice that the flies pace back and forth from one end of the hallway to the other. That's actually convenient because they sample then space pretty reliably. And you'll notice that when the stimulus epoch begins, the flies tend to slow down. And importantly, when this fly encounters the boundary between the odor and non-odorized part of the hallway, it tends to turn around and head back into the odorized part of the hallway. And it turns out that this turning round behavior is crucial. This slowing is a reliable feature, but it turns out not to be spatially specific. So they tend to walk slower throughout the apparatus during the stimulus epoch and for some time afterwards. So here is just an example, again, randomly selected small number of traces aligned to the point of odor entry. And here's some more trajectories aligned to the point of odor exit. And what you should be comparing this to are similar trajectories when the fly is walking through the same part of the chamber, but when there's no blue light present at all. And what you can see is that blue light entry has little discernible effect on the fly's behavior, but blue light exit, fictive odor exit, has a marked effect where they tend to turn around and re-enter the odor at a higher than normal probability. So here you can just see average trajectories, now averaged across all flies and separated into categories when they're entering and exiting the odor. And you can see on average there's really little to no effect of odor entry, but there's a substantial effect of odor exit. They turn around to re-enter the odor. Keep in mind that this is a probabilistic behavior, and even at maximum light intensity, the fly is not reliably re-entering the odor. But it is doing so at a rate very substantially higher than it does in the absence of any odor. So we can quantify this behavior using a simple metric. We define a choice zone at the middle of the apparatus, and we just count the exits into the odor versus the exits into the non-odorized sector. And we derive this preference index, again, adopted from the Miesenbach lab's previous publications. And what you'll notice is that this preference index rises monotonically as a function of blue light intensity. And in this case, where most ORNs express channel rhodopsin, the maximum value of this preference index is close to 0.6. And that corresponds to flies choosing the odorized side about 4 times more often than they choose the non-odorized side. So we get a pretty robust behavior in flies that don't express Gal4. There's no preference, and that's the result of us engineering that situation to exist. So I want to now tell you something that was very surprising to us when we stumbled across it. You'll remember that air flows into the chamber at both ends, and it flows out at the midpoint. That was a historical artifact. One day we turned off the air, and we found that the behavioral preference for the odor was abolished. So, and this was surprising to us. In this case, there's really no preference, either in terms of occupancy or in terms of a turning bias. You'll just see when you compare the black and gray trajectories that there's a slight increase in walking speed as a result of the blue light, but it doesn't produce any preference for the blue side. And here's just the preference index as a function of blue light intensity. It's essentially flat when there's no airflow in the chamber. And so this suggested 2 interesting alternative hypotheses to us. One possibility is that airflow is simply permissive. So in that case, the fly's turning direction, its walking direction, is guided by odor cues, but for some reason, wind is permissive for those decisions. An alternative hypothesis is the fly's turning, its walking is guided by the wind vector, and that the presence of odor is simply permissive. So to tell the difference between those 2 alternatives, we focused on the beginning of the stimulus epoch, because that's a time when wind direction and odor are dissociated. So what happens is the fly can be walking in either direction in one half of the chamber, the half that's destined to be blue. And when we turn on the blue light, we can catch it either walking upwind or walking downwind. So we divide the trajectories according to the walking direction at trial onset. And what we see is that when the fly happens to be walking upwind when the blue light comes on, there's really no discernible effect on its walking trajectory. But when it's walking downwind, there's a strong tendency for it to reverse and head back upwind at odor onset. So I think this is a strong indication that the fly's walking direction is not guided by odor, it's guided by wind direction, and odor somehow releases or gates that behavior. We can also look at the offset of the trial. When the stimulus turns off, when the blue laser turns off, and the fly happens to be walking upwind, there's a statistically significant tendency for it to turn and head back downwind. But when the fly happens to be walking downwind when the odor comes off, it turns out that there's no significant difference here in its walking trajectory. So this suggests two pretty simple rules, which together could account for all the behavioral data that I've shown you so far. Rule 1, at odor onset, walk upwind. And rule 2, at odor offset, walk downwind. And when you think about it, those are actually really useful rules to have in nature, because in nature, odors tend to move downwind from their source, okay? So if you want to find the source of an attractive odor, fly or walk upwind. That makes sense. Conversely, if you are moving upwind and you lose the odor, it may be that you've overshot the odor source, or you've simply moved out of the turbulent plume, in which case reversing course and walking downwind again is a useful way to pick up the odor plume once more. So we can actually, it's quite difficult to put real odors downwind instead of upwind, but we can do that with optogenetic odors. And that provides more evidence the direction of behavior depends on the odor-wind relationship. So here's the arrangement of airflow in our conventional apparatus I've been telling you about. But we can repipe this apparatus so that air just flows in one direction. And we can place the fictive odor downwind, so to speak. And that produces a very whimsical behavior in the flies. They tend to exit the odor, turn around to head back in. Then they enter the odor and they turn around to head back out. And they'll do this all day, 5 hours at a time, and they'll never learn. I think a previous speaker called the fly brain today minimal, and I think this might be evidence for that minimality. But really, this strange behavior follows directly from the two rules that I just told you about. So rule 1, walk upwind in response to odor onset. Well, that's exactly what's going on here. The flies enter the odor, and then they walk upwind, which involves a reversal. All right, rule 2, walk downwind at odor offset. Well, here the flies exit the odor, and then they walk downwind. So I think that this is pretty clear evidence, again, that what we're looking at here is an odor-gated or odor-released behavior, which is guided by the direction of the wind vector. So now I'm going to turn to the question of how this behavior depends on ORN activity. That's the question I really posed to you at the beginning of the talk. So we're going to return to our conventional apparatus here because it produces a really robust preference in flies where most ORNs are activated by channel rhodopsin. And in every experiment, we're going to use 8 different blue light intensities, each repeated many times, randomly interleaved. That corresponds to about 5 hours of data per fly. In this study as a whole, we looked at 60 different genotypes, so defining different combinations of ORN activity. This gives you a sense of why this study would be hard to do in flies. It would cost $1 zillion. We looked at almost 3,000 flies in total for a total of more than 15,000 hours of data. So a big undertaking. So here I'm showing you behavior elicited by single olfactory channel. So in these experiments, we're driving channel rhodopsin expression in a single ORN type, all the ORNs presynaptic to one glomerulus, each glomerulus here labeled in the upper left-hand corner. I'm plotting behavior, that is the preference index, as a function of the measured ORN firing rate for those corresponding ORNs in the same genotype. And the shaded regions here represent bivariate 95% confidence intervals computed using a bootstrap approach. So we were able to measure ORN activity for 6 of the glomeruli I'm going to show you on this slide. For the remaining 2 glomeruli, the ORNs were not accessible due to their location on the antenna. So I'm just plotting behavior as a function of blue light intensity in these 2 cases. And these are, again, 95% confidence intervals. So I want you to notice a few things. All 8 of these glomeruli are driving behavioral attraction. Later in the talk, I'll mention a case where we witnessed behavioral aversion for a different glomerulus, but for now I'm going to focus on these 8 attractive glomeruli and their combinations. You'll notice that there's consistently a monotonic relationship between ORN firing rate and behavior. We never observed a non-monotonic relationship, actually. You'll notice that all the ORNs have non-zero spontaneous firing rates. That's why these curves start at some non-zero value on the x-axis. And that a relatively small increase in ORN firing rate of about 5 or 10 hertz is generally sufficient to evoke some measurable behavior. That's interesting because the maximum odor-evoked firing rate for these neurons is about 300 hertz. So what we're looking at here, it really in total, is the bottom 10% of their dynamic range. And a really tiny change in their overall possible firing rate change is what's sufficient to produce a behavior. And that's important because in our field of Drosophila olfaction, often ORN firing rates below 50 hertz are scored as 0, literally. So what this tells us is that small ORN firing rates are relevant. Interestingly, behavior tends to saturate in most of these cases at pretty low ORN firing rates. And that's notable because you'll recall that the maximum preference index that we can record is something closer to 0.6. And so this is clearly not some limitation on the animal's behavior. There's something in the brain that's limiting the effect of single processing channels. So what happens when we combine these channels? So I'm going to begin just slowly building up here on one pair of glomeruli. They are called DM1 and VA2. These are not very user-friendly names. I'm sorry about that. Here I'm showing you behavioral responses to stimulating these 2 glomeruli individually. And in gray, you'll see the response to stimulating them together in the same fly. And what you'll notice is that the behavioral response in this case looks quite a lot like the predicted linear sum of the 2 component responses. And this indeed is what the dominant model in the field right now would predict. If each glomerulus makes an independent and fixed contribution to behavior, then the response to co-stimulation should look like the sum. So now let's look at VA2 again, but this time combined with a different glomerulus, DM2. Again, now the combination response looks a lot like the sum. But now let's compare the orange glomerulus, DM1, with the purple glomerulus, DM2. And you'll notice that in this case, these 2 glomeruli do not summate. The response to the combination looks a lot like the response to the more potent of the 2 component glomeruli. And I think this is a notable negative result, the failure to see summation here, because, again, recall that each of these 2 glomeruli, DM1 and DM2, summated effectively with VA2, all right, but they don't summate effectively with each other. And that suggests there's something in the way that these glomeruli are wired up in the brain that creates an antagonistic relationship between them, such that co-stimulation reduces the efficacy of one or both glomeruli. And in fact, this result is a little bit suggestive of a winner-take-all situation. So we can summarize these results using a simple network diagram. We imagine these 2 glomeruli connecting to some higher brain region such that their effects summate. We can call that a summing node. These 2 glomeruli, DM2 and VA2, also effectively sum. The outputs of these nodes then lead to the behavioral preference. But we have to imagine there's some kind of antagonism, perhaps at the level of these summing nodes, that accounts for why co-stimulation of the purple and the orange glomerulus seems to reduce the effect of one or both glomeruli. So now we can extend this exercise to all pairwise combinations of the 8 glomeruli I showed you. I'm going to show you a lot of data at once. So just to orient you again, I'm going to be showing preference index as a function of log blue light intensity. Here's the data for one glomerulus, DM2. Here's this data reproduced many times. We're going to look at this in combination with all these other glomeruli. In blue, you're looking at the singleton responses to the blue glomeruli. And in black, you're looking at the combination responses. And I'm going to indicate with magenta asterisks here cases where the combination response is statistically significantly smaller than the predicted component sum, again, using a bootstrapping approach. And I'm going to be showing you the results of 28 independent statistical tests. So of course, we had to do an appropriate multiple comparisons correction to deal with that issue. So here you can see data for all possible pairwise combinations of the 8 attractive glomeruli in our data set. And it turns out that in 5 cases, the response to the combination was significantly smaller than the predicted linear sum, even with the multiple comparisons correction. So I think this phenomenon of antagonism is clearly not a limited phenomenon. What you'll notice is that antagonism is not characteristic of any one glomerulus. It's not a property of the glomerulus in particular. It doesn't dominate one row or one column of this matrix. Instead, it's a property of the identity of both glomeruli in the combination. So we can summarize these results in a binary fashion. Magenta squares are cases where the combination was significantly smaller than the predicted linear sum. And in green pertains to cases where there was no significant difference from the predicted sum. So we can summarize these data in another way by extending our notion of a network diagram to include all of these results. And we draw this diagram just using the rule that I described to you before. If 2 glomeruli summate, we draw them as connecting to a common node. And if 2 glomeruli do not summate, we draw them as connecting to different nodes. And then we use the minimum number of nodes that we need to explain all the data. And this procedure does not mathematically need to give rise to a unique solution, but in our case, it did give rise to a unique network diagram that describes the behavioral data that I just showed you. I just want to make the point that although this diagram looks very black and white, quite literally, we have more confidence in some edges of this diagram than in other edges, okay? And one way to think about this is to look critically at the assumptions that went into the hypothesis testing that we used to construct the diagram. So recall that our null hypothesis is that glomeruli will summate. I think that's a reasonable null hypothesis because that's the dominant idea in the field. Each glomerulus, according to this model, makes an independent fixed contribution to behavior. And so we required strong evidence, p less than 0.05, in order to conclude that there was antagonism and to shape the network diagram accordingly. But we can turn this assumption on its head and we can say, all right, let's imagine the null hypothesis is antagonism. Every glomerulus antagonizes all other glomeruli. And then in that case, we require strong evidence, again, p less than 0.05, to classify a pair of glomeruli as actually summing. And so we can then think about each pairwise combination in terms of the likelihood ratio for those 2 tests, the probability of summation divided by the probability of antagonism. What I want you to notice is that there are some entries in this matrix, again, that are strongly magenta. Those are the cases where, again, we have significant evidence for antagonism. There are other entries in this matrix that are strongly green. This is a case where we have very significant evidence for summation as compared to the hypothesis of antagonism. I think the entries in the matrix indicated with strong colors are the ones that we should have confidence in the network diagram. And the entries that are marked with pale colors are the ones that we just don't have strong evidence for either way. I think the point here is not to make a detailed inspection of all the edges in this diagram, but instead, I think what we can have confidence in is the general statement that there's not a single rule that predicts how all glomeruli will combine. Rather, some glomeruli appear to summate and others appear to antagonize each other. So the big picture then is that the response to the pair in some cases is significantly larger than the response to the larger singleton. In other cases, some glomeruli antagonize each other. The response to the pair is significantly smaller than the sum of the singleton responses. And so although we may have uncertainty about some of these combinations and how we ought to categorize them, I think we have confidence in the general statement that glomerular interactions can be divided into 2 categories, summation and antagonism. And given this, I think these data imply that groups of glomeruli compete for control of the behavior. So what about aversion? I mean, carbon dioxide is an example of an odorant which is generally aversive to Drosophila. This is illustrated, for example, by nice data from Marine de Bruyne's lab. Here are walking trajectories in a two-dimensional chamber. If you infuse carbon dioxide into one end of the chamber, the flies avoid it. And it turns out that carbon dioxide quite specifically activates one ORN type. That was shown by David Anderson's lab and John Carlson's lab initially. Here's just functional imaging data from my lab more recently, making this point clearly. These are different planes sectioning through the antennal lobe, so the whole array of glomeruli. And you can see that carbon dioxide evokes activity quite specifically in one glomerulus, and that's glomerulus V. So it turns out that if we drive activity in the V ORNs, that combined with the orco population, that is a large majority of ORN types that express the orco gene, that is sufficient to very substantially diminish the behavioral attraction elicited by that fictive odor stimulus. And that's notable because this is only one glomerulus pitted against an array of something like 30 some odd glomeruli. Right? So this is a potent vote against attraction. And when we examined this in detail, when we combined the V glomerulus with every one of our individual attractive glomeruli, we saw a statistically significant suppression of attraction. So we might update the network diagram then to imagine that V is antagonizing the effect of these glomeruli at an early stage or at a late stage of integration. So the last question I'm going to ask in my allotted time, which is apparently drawing to a close, is are there other interesting behaviors in our data? So we can make a list of other behaviors reportedly modulated by odor in walking insects. This list owes a lot to the work of the late British entomologist J.S. Kennedy. Basically, these behavioral parameters consist of measurements of heading angle, speed, probability of stopping, and sideways speed, triggered on various relevant odor encounters. We can compute a signal to noise for each behavior just to ask whether or not it's reliably modulated. Basically, the signal to noise metric expresses the variation in this measure across experimental conditions, that is across genotypes or laser powers, divided by the variability within an experimental condition. And what you'll see is that the most reliable of these parameters by far is the heading after odor exit. And that shouldn't surprise you based on what I told you before, that most of the behavior that's going on in our chamber that's modulated reliably by odors is this exit turnaround behavior. So what we really want to know then is basically, is odor modulating just one thing, one behavioral program, or is it modulating these parameters independently? To ask this, we can just reduce the dimensionality of the data using principal components analysis, and it turns out that there are only two significant axes in this behavior space. So now we're going to look at our data in the two-dimensional space defined by PC1 and PC2, which together account for almost all the explainable variance in the data. Here, each color represents a different genotype. This is orco, most ORNs express channelrhodopsin, and this thick blue line represents control flies where no channelrhodopsin is expressed. And each point along this line represents a different laser power. This is 0 laser power, and then the lines diverge as laser power increases. And what you'll notice is that laser power is doing something in control flies. We felt kind of miffed about this because we'd worked hard to engineer that away. But the nice thing about this analysis is it's revealed a previously unsuspected feature of behavior to us. What you'll notice is that, however, all the control behavior lies along one line, one axis in this space. And we're going to call this the thermal axis because we think it reflects thermal cues. And you'll see that the orthogonal axis, which we call the odor axis, accounts for almost all the behavior that depends on channelrhodopsin expression that's present in the channelrhodopsin expressing flies and that varies as a function of genotype. So we can look at behavior along the thermal axis as a function of blue light intensity. To a first approximation, it's pretty similar for all the flies. It turns out to basically just represent changes in walking speed. But this odor axis turns out to capture almost all the effects of light that depend on channelrhodopsin. I've color-coded genotypes according to the rank order of their maximum preference index. And it turns out that this axis correlates really strongly with the preference index metric. So it turns out the preference index is almost the whole ballgame here. So just to summarize this, it turns out that many behavioral parameters are modulated by our fictive odor stimuli. but virtually all the behavioral variation that depends on chenorodopsin expression is captured by one axis, which is also highly correlated with the preference index. And this means these behaviors are modulated in a highly correlated manner, not independently, and so they represent a coherent behavioral program. And this program largely consists of downwind walking at odor onset and upwind walking at odor onset. So I'm going to conclude by articulating what I think the big picture conclusions are that come from this study. First, we've defined a clear behavioral program. Odor onset triggers upwind walking, and odor offset triggers downwind walking. The frequency of this behavioral program depends on ORN identity and ORN firing rate, not surprisingly. And I think what was surprising to us is that it looks as if groups of glomeruli are competing for control of this behavioral program. And more specifically, our results, the network diagram we've deduced, begins to define the glomerular membership of these groups. Some glomeruli antagonize this behavioral program, notably glomerulus V. And finally, the elemental behaviors that comprise this program turn out to be modulated by odors in a correlated manner. Really, that's a tautology. By definition, that's what a behavioral program is. But I want to say this out loud because I think what we've done is to show in a careful way that that's pretty rigorously true in this data set. So future directions for our lab and for the field come out of this study. First, what are the circuit mechanisms that allow odor onset and offset to gate upwind and downwind walking respectively? David Tank drew attention in his talk to the notion of a contingency switching task, which is thought to require the cortex in mammals. You could think of this as a kind of a primitive contingency switching. Really, the fly's walking direction relative to the odor vector is switched depending on whether the odor concentration is increasing or decreasing. And so we'd like to understand how that occurs in a very simple brain. What constraints limit the behavioral effect of activating a single glomerulus? Why is behavior so noisy, not just near threshold, but also near saturation? And why does behavior saturate? How are summation and antagonism implemented and where? What are the neural circuits that might correspond to these algorithms? And what is special about the membership of the groups of glomeruli that appear to compete for control of behavior? We hypothesize that glomeruli are grouped together based on the statistical likelihood of their co-activation by either common odor stimuli or behaviorally salient odor stimuli, and that should be a testable hypothesis. So I just want to close by pointing out that all of the work in this study is the work of a single graduate student, Joe Bell, who's actually an MD PhD student. And the MD PhD office at Harvard asked him, after he'd been in the lab for 4 years, why he was doing a particularly long PhD. And they asked him again after 5 years why he was doing an even longer PhD. And after 6 years, they asked him if he would please leave his PhD lab. And to Joe's credit, he applied the most extraordinary amount of intelligence and commitment to this project so that he actually became an inspiration to me and to the other lab members, and I salute him for that. Thanks, too, for the people who gave us flies, helped us with math, gave us advice, and gave us funding. Thank you.
For more, please visit us at stanford.edu.